Introduction. Welcome to today's lecture on the topic of threonine and its various biochemical pathways and functions. Threonine is one of the 20 essential amino acids that make up proteins. It plays a crucial role in various metabolic processes, as well as being involved in the synthesis of important molecules such as neurotransmitters and lipids. In this lecture, we will explore the enzymes and pathways related to threonine metabolism, the significance of threonine-derived metabolites, and how alterations in threonine metabolism can contribute to certain diseases. So, let's dive into the fascinating world of threonine biochemistry. Threonine dehydrogenase is an enzyme that catalyzes the conversion of threonine to 2-amino-3-ketobutyrate. This reaction involves the removal of a hydrogen atom from threonine, resulting in the formation of a keto group on the carbon adjacent to the amino group. Threonine dehydrogenase is essential in both prokaryotes and eukaryotes for the breakdown of threonine and its utilization as a carbon and nitrogen source. The product of threonine dehydrogenase, 2-amino-3-ketobutyrate, can further enter several metabolic pathways. Threonine transaminase, also known as threonine aminotransferase, is an enzyme that catalyzes the reversible transfer of an amino group from threonine to alpha-ketoglutarate, forming glycine and 2-ketobutyrate. This enzyme is part of the larger family of aminotransferases, which play a vital role in amino acid metabolism by facilitating the interconversion of different amino acids. Threonine transaminase acts as a key player in the catabolic pathway of threonine degradation, contributing to the synthesis of glycine and other important metabolites. Threonine aldolase, also referred to as threonine aldolase I, is an enzyme that catalyzes the reversible cleavage of threonine into glycine and acetaldehyde. This reaction is a crucial step in the degradation pathway of threonine. Threonine aldolase has been identified in various organisms, including bacteria, fungi, plants, and animals. Interestingly, threonine aldolase II, a distinct enzyme found in certain bacteria, catalyzes the conversion of threonine to glycine and acetyl-CoA, highlighting the diversity of the threonine degradation pathway. Threonine phosphorylation refers to the addition of a phosphate group to the hydroxyl side chain of threonine. This post-translational modification is typically catalyzed by kinases, which play a central role in signal transduction pathways, cell cycle regulation, and various cellular processes. Threonine phosphorylation is an essential regulatory mechanism in the activation or inhibition of proteins involved in cell signaling and metabolism. This modification can modulate protein structure and function, leading to diverse cellular responses. Threonine hydroxymethyltransferase is an enzyme that catalyzes the reversible conversion of threonine to glycine, while generating formaldehyde as a byproduct. This enzyme plays a crucial role in the interconversion of threonine and glycine, as well as folate metabolism. Threonine hydroxymethyltransferase is particularly important during embryonic development and in rapidly dividing cells, where it contributes to the synthesis of glycine, a key component of nucleic acids, proteins, and other cellular components. Threonine acetyltransferase is an enzyme involved in the acetylation of threonine residues in proteins. This modification can alter protein stability, subcellular localization, and interaction with other molecules. Threonine acetyltransferase activity has been observed in various organisms, including bacteria, yeast, and mammals, highlighting its evolutionary conservation and potential role in protein regulation. Further research is needed to fully understand the functional significance of threonine acetylation in cellular processes. Threonine synthase is an enzyme that catalyzes the final step in the biosynthesis of threonine from homoserine. This reaction involves the conversion of homoserine to threonine in the presence of pyridoxal phosphate, PLP, as a cofactor. Threonine synthase is an essential enzyme in organisms capable of de novo synthesis of threonine. The regulation of threonine synthase activity is tightly controlled to maintain appropriate levels of threonine for protein synthesis and metabolic processes. Threonine degradation refers to the breakdown of threonine into various metabolic intermediates, eventually leading to the production of energy and the utilization of carbon and nitrogen sources. The threonine degradation pathway involves a series of enzymatic reactions, including dehydrogenation, transamination, and cleavage. This pathway contributes to the synthesis of glycine, acetyl-CoA, and other important metabolites required for various cellular processes. Threonine serves as a precursor for several important metabolites in the body. For instance, threonine is converted into glycine, which participates in protein synthesis, DNA synthesis, and neurotransmission. Additionally, threonine is a precursor for the synthesis of serine, an amino acid involved in nucleotide metabolism and phospholipid synthesis. The interplay between threonine and its derived metabolites plays a critical role in maintaining cellular homeostasis and supporting vital physiological functions. Alterations in threonine metabolism can have significant implications for human health, contributing to the development of various diseases. For example, deficiencies in enzymes involved in threonine degradation or biosynthesis can lead to inborn errors of metabolism, characterized by neurological and developmental abnormalities. Moreover, dysregulation of threonine availability or metabolism has also been implicated in conditions such as cancer, cardiovascular disease, and neurological disorders. Understanding the role of threonine in disease pathogenesis is essential for developing targeted therapies. Threonine plays a vital role in neurotransmitter synthesis, particularly in the production of glycine and gamma-aminobutyric acid, GABA. Glycine acts as an inhibitory neurotransmitter in the central nervous system, modulating neuronal excitability, whereas GABA is the main inhibitory neurotransmitter. Threonine-derived glycine and serine serve as precursors for the synthesis of these important neurotransmitters, highlighting the link between threonine metabolism and neurological function. Threonine contributes to lipid metabolism through various mechanisms. It is involved in the synthesis of sphingolipids, a class of lipids essential for the structure and function of cell membranes. Threonine serves as a precursor for the amino acid cysteine, which, in turn, is required for the synthesis of glutathione, a crucial antioxidant involved in lipid metabolism. Furthermore, alterations in threonine availability or metabolism can impact lipid homeostasis and contribute to metabolic disorders such as obesity and dyslipidemia. In conclusion, threonine is a multifaceted amino acid that plays a pivotal role in diverse biochemical pathways and cellular functions. Its metabolism involves a network of enzymes, including threonine dehydrogenase, transaminase, and synthase, which contribute to its degradation, biosynthesis, and utilization in various metabolic processes. Threonine-derived metabolites participate in the synthesis of important molecules such as neurotransmitters and sphingolipids, while dysregulation of threonine metabolism can contribute to the development of diseases. Understanding threonine's role in cellular physiology and pathophysiology is vital for advancing our knowledge of human biology and developing targeted therapeutic interventions.